everyone, welcome to Marine Microbial Ecology. This is the introductory lecture on phytoplankton ecology. Let's get started. So we're dealing with plankton. Plankton has the same Greek root planktos as planets, so these are things that drift or wander. They're small organisms, usually. They have very little or limited power of locomotion, and they just float and drift with the water movement. They can't overcome tides or currents. Some of them do have the power of locomotion. They have flagella, they have cilia. Some of them can beat rhythmically like jellyfish, but generally they can't overcome the currents, the tides. They have to go where they're taken. The chap in the top left hand corner there, that's Victor Hensen. He led a German expedition through the North and South Atlantic back in 1889, and he's basically considered the father of biological oceanography. So when you hear people like me talk about algae, phytoplankton, microplankton, we tend to use these terms interchangeably, which isn't quite right, but it happens all the time. So algae, the way they're referred to most commonly, is just a general name, which is no taxonomic relevance really. It's just a catch-all that kind of takes things from tiny, small, little unicellular organisms all the way up to algae like seaweeds and giant kelps. So I'm mainly interested in these little guys, the microplankton. They're between 20 and 200 microns in size. And they come from every taxa you can imagine. So they're really diverse. They're diverse in terms of their size, of their shape, of their way of living. They have masses of different ways of interacting with their environment. So these are just pictures here, all taken on the scanning and electron microscope down in SAMS. So dinoflagellates, they come from the Greek dinos, which in this case means whirling, and the Latin for whip, which is flagellum. So there's about 1,500 species of these described in the literature. They're unicellular. Some of them are autotrophic, so they photosynthesize, create their food from light. Some of them are heterotrophic. They eat other little beasties, and what we're finding now, week by week, we find papers coming out that say what we thought were formerly heterotrophic species are actually mixotrophic. So in this case they can actually grow their own chloroplasts, or more likely they steal them from other organisms. So they go along, they ingest some other organism, they steal the chloroplasts, they distribute it around their cell, and then they use that to photosynthesize. And often it can last for 2 or 3 months before these chloroplasts die off, and then they go off and find some more to steal. Diatoms, they're really common. They're autotrophs. They've been around at least 180 million years. We find their fossils in the Jurassic period. There's about 100,000 different species of these. Again, they're unicellular, but often they'll live in colonies. And up in the top right hand corner there you see a small chain of Chaetoceros, and there's actually 3 or 4 cells there joined together. They have these setae, spines that stick out, and this kind of links the cells together, and it's also a distinctive way to identify different species. So these guys live in little silica boxes, frustules we call them. They have 2 overlapping sections, and if you imagine one of those plastic petri dishes from the lab, how the top fits over the bottom, that's pretty much what these diatoms do. Now this box can be twisted and contorted into all different shapes and have spines attached to it, but basically that's the idea. It's just like a petri box, one lid on top of the other. So the top is the epitheca and the bottom is the hypotheca. Around the middle they have a series of girdle bands, or pleura, and these allow the cells to grow and also help when they have to reproduce. And these guys can reproduce asexually. They just split a half. You get 2 cells, these split a half, so they can grow exponentially. They can grow very quickly when conditions are right. We also have things like the nanoplankton, between 2 and 20 micrometers. They're often tiny little flagellates. Some of these can cause problems for fish. And even smaller, we have the picoplankton, which can be less than 2 micrometers. In this case, the picture is of Synococcus, which is a cyanobacteria that's common in the sea. So why should we care about these little guys? Well, phytoplankton is responsible for about 50% of the primary production on the planet. So 50% of the food on the planet comes from these organisms in the sea. They also produce about 50% of the oxygen we breathe, and they draw down about 50% of the atmospheric CO2 we pump into the atmosphere. So if you look at the map there, you can see, if you download this PowerPoint presentation, that's actually an animation, and you'll see as the globe revolves that the deep sea, the big oceanic gyres, tend to be blue, lacking in very much chlorophyll, whereas around the coastlines, or where, for example, the Amazon jets out into the Atlantic, you see high concentrations of chlorophyll as a proxy for phytoplankton. So this is where they tend to grow, around the ocean, the coastal margins. Another important role they play is in the marine food web. So they're at the base of pretty much every marine food web we can imagine. Let me just click on a few slides here. So they go from the smallest crustaceans or anemones all the way up to things like the large baleen whales. So they're incredibly important when it comes to food in the sea. They're almost like the grass of the sea. Let me just click on through the slides. And obviously at the top of the marine food web, the one we care about is us. So we take an awful lot of our protein from the sea. Another reason they're important is something we call the biological pump. So here's a simplified diagram of it. It can be a little bit more complicated than that, and depending on where and when it happens, this can actually put more carbon dioxide back into the air. But in general, these phytoplankton are fixing carbon from the atmosphere, in turn, and producing oxygen. In turn, they're grazed by zooplankton, by ciliates and heterotrophic dinoflagellates. And as they break up, a lot of their nutrients are remineralized by bacteria in the water column. And this is called the microbial loop, and we'll cover this in other lectures later on. So as these particles are broken down, some of them, like I said, most of them are recycled, but some of them fall down to the sediments, where they're buried and end up turning back into rock and going into geological processes. So this is constantly drawing carbon dioxide down from the atmosphere. How would you look after your plankton? So you start off with a sad little plankton, you provide them with plenty of light, a few nutrients, things like phosphorus and nitrogen, silicon and iron, and you'll have a happy little guy. So these next few slides just are diagrams of the different marine nutrient cycles. So we've got nitrogen, phosphorus and silicon, and you can pause this presentation or download the one on Brightspace and go over these at your leisure. So how are the nutrients distributed in the sea? So these are three vertical profiles. We've got the depth down the left hand side and the nutrient along the top. So we've got phosphate on the left, nitrate in the middle, silicate on the right. And this is how they're distributed. The solid lines are for the Atlantic and the dotted line is for the Pacific. So you can think for a couple of minutes why we might find more nutrients in the Pacific than we do in the Atlantic. But what's obvious from these diagrams is that up in the surface waters, these nutrients are in short supply, and this is because they're being used up by the biology. So the phytoplankton are sucking up these things to help them grow. As you go deeper in the water column, the nutrients, because the sediments are constantly settling, the nutrients increase in concentration. When we get winter storms or big turnover events, this will bring nutrients back up into the water column. But generally the high concentrations are at the bottom. So why are they different in the Atlantic and the Pacific? Let's look at the next slide. So it's due to the thermohaline circulation. So surface waters flow up to the poles. There they get chilled, they become quite rich in oxygen. They also become very dense, and they sink down onto the bottom of the ocean basins and flow back towards the equator. So they have this complicated looping system around the globe. And basically what happens is that a lot of the nutrients get carried into the Pacific, and that's where the water rises up into the surface layers, where it then carries on up to the poles and sinks back down again. So a lot of nutrients get dumped in the Pacific, and that's why the nutrient concentrations are higher there. Another really important thing we have to think about is the thermocline. So the thermocline is just a change in temperature in the water masses. So what we tend to find is we've got this deep, quite saline, oxygen-rich, nutrient-rich water, which is dense, and then it's topped by a shallow mixed layer of less dense or less saline water on the top, often warmer. So if you look at the temperature, the mixed layer will be warmer. Then you've got a transition zone, which we call the thermocline, before it hits the really cold bottom water. And you may have come across this if you've been out snorkeling or diving, even just swimming, and you're in nice warm surface waters, and you see a bit of coral or a fish you want to dive down to, and as you go down you just feel the water get much colder. You seem to go through a transition, and this would be the thermocline. Now in winter, this top mixed layer expands, it gets much deeper. Storms and winds, they overturn the water column, so you've often still got a deep cold layer with a much shallower thermocline and then a large mixed layer, and often there's very little difference between the temperature, and depending where you are, this temperature profile can be vertical all the way down through the water column, so you just have this mixed layer. So here's some actual plots that I took at the Craig Isles, which are just off Lismore. You can sort of see them at the back of the lab. So I've got the temperature profile, I've got the density in the middle, and I've got chlorophyll, which is the proxy for phytoplankton. So if I drop a line down on here, you can see that they line up incredibly well. So you've got warm temperature in the area above the dotted line, which corresponds with low density water, and that corresponds with a high chlorophyll maximum. And then below that we've got colder temperatures, higher density, and the chlorophyll falls off. And the reason the chlorophyll or the phytoplankton tend to gather around this picnic line or thermocline is because they've got this constant battle between the light from the top and nutrients seeping up from underneath. So here's a profile. This is a surfer profile from, this is the Linn of Lorne in Loch Crieran in this diagram at the back there. You can see the temperature profile there. So again, we've got much warmer temperatures at the top and colder waters at the bottom. If you look at the graph in the background, you can see that there's a sort of wedge of high density salty water pushing up the Linn of Lorne. Then the first peak is the sill into Crieran, and then as you move up towards the right hand side, you can see this surface layer of much, it's warmer and less saline water, and this is stratified due to the fresh water flowing in from River Crieran. You also see a little bit of less stratified water, less dense water flowing down on the left hand side of the screen, and this is due to water from Loch Etive. So it pours out of Loch Etive, it hits the big bay there, and then the fresh water flows, or the brackish water flows across to the Craig Isles. So that you have to be aware that the axes there are incredibly exaggerated. So we've got 50 meters from top to bottom and 18 kilometers from left to right. So we don't actually have these big jagged peaks out in the Linn of Lorne or the Loch Crieran. These are just gentle bumps and rises in the seabed. So another important thing that affects phytoplankton production, primary productivity, is the Coriolis force. So in the northern hemisphere, anything moving across the surface tends to get moved to the right because of the Coriolis effect. So you'll have covered this in oceanography lectures, I'm sure. It's a little bit if you're on a playground, merry-go-round in a school playground, and you start it spinning and you try to walk across it, you get thrown off to the right hand side. In the southern hemisphere it's the opposite, things flow to the left. So if you look at the first diagram on the left, we've got wind which starts to push the water in one direction. The water's got a certain amount of friction, inertia, it doesn't want to move, so it drags backwards, and all the time the Coriolis effect is pushing this water down to the right. So as things progress, the water starts to flow off at an angle, the Coriolis force keeps turning round and balances with the water drag, and finally the water takes up a position where it's flowing at about 45 degrees to the prevailing wind direction. So that's very simplified the way it works, but generally if you've got a wind in one direction, the water's flowing at about 45 degrees to the right of it. So what does this mean for phytoplankton production? Well, if you take a situation like this, so if you imagine the wind's blowing out of the top of the page and the coastline's on the right there, because it's going to move the water off towards the right, and call this Ekman drift, the surface waters are going to get pushed away to the right and leave space basically. And this will be filmed by cold, deeper, nutrient-rich water upwelling from underneath. So this nutrient-rich water helps to feed a lot of phytoplankton. And this is why, if you think back to that first map or the animation of the globe, you'd see all the chlorophyll was gathered around the coastal zones and not out in the open gyres. This is why, because that's where the nutrients are coming up. So if we consider one of the big fisheries in Peru, so this is a simplified diagram of what happens off Peru, and there they have incredibly rich fisheries, and this is because they have these huge blooms of phytoplankton. So again, you've got offshore winds that are blowing the surface waters out to sea. This leaves a gap for this nutrient-rich water to upwell into the coastal zones. Any phytoplankton that are caught in these surface layers, they've got plenty of nutrients, there's plenty of light, so they start to grow, and they grow exponentially. By the time you get 18 kilometers offshore, we've got massive blooms of phytoplankton, and this is feeding the fish, and it supports a huge fishery. By the time you get out to about 30, 60 kilometers, most of the nutrients in the surface layer have been used up, the bloom starts to crash and die, and it sinks back down, and the whole cycle starts all over again. Now, in the equator, this isn't quite the case. There's no Coriolis force at the equator, but to either side we do have the trade winds blowing from the west. So this moves the water to the right in the north hemisphere, moves it to the left in the southern hemisphere, and this causes cold water to upwell from 100 meters deep or so, bringing nutrients up into the surface waters. So if you look on any maps, you will see that there's a sort of plume that runs along the equator where you do get some primary productivity. And finally, in this talk, I'll just cover tidal fronts. So this is anywhere tides tend to flow in and out of an area. It could be a bay, it could be in a strait or whatever. So basically you'll get high nutrient cold water on the bottom, and if it's running up ashore, if the bathymetry is getting shallower, as there's friction with the bottom of the sea, it starts to cause turbulence. So nutrients get brought up from these bottom sediments. On the top you've maybe got stratified water, and then inland you'll have the mixed water, and where these meet we call this a tidal front. So nutrients are constantly seeping up between this deep nutrient-rich layer, which is very turbulent, into the upper stratified layers. And as it reaches the mixed water, again turbulence causes this vertical diffusion of nutrients from the top to the bottom. So tidal fronts are also places where we get high numbers of phytoplankton and good primary productivity. So this is just a sort of diagram showing, this is the southwest of Iceland, Faxaflói Bay, and it's just showing the progression of a phytoplankton bloom. So if you start off up on the left, early in March, there's not much going on, but there'll be fresh water flowing out into this bay. By the end of March, because the water is stratified, because it's fresh, it's flowing on the dense water, and the light's beginning to increase, we start to see the growth of phytoplankton, and that's shown by the little dark mark inside the bay. By April, it's beginning to spread out and around the coastal zones, and by May, there's a full bloom in progress, and you can see high concentrations of phytoplankton all around the southwest coast. Now, as we said, they're going to start using up the nutrients. They'll use these nutrients up very quickly, and by the end of May, the nutrients are gone and the phytoplankton have already started to crash and die off. And this is quite typical. So this illustration is just showing the relationship between the mixing depth and the photic zone. So the photic zone is the area we consider the usable light gets down into the water, how deep it gets into the water column. So if the photic zone and the mixing depth are similar, it means any phytoplankton in this upper mixed layer will be circulating around, and it'll always be in the light, so we'll get good growth. There's plenty of light for them to grow, and as long as there's sufficient nutrients, they'll bloom, they'll grow quite easily. If the situation changes, so through storms, through situations in the early winter, spring, that the mixing depth is actually deeper than the photic zone, then the phytoplankton are going to spend part of their time down in the dark, where they won't photosynthesize, and this will slow down primary productivity. So we're always very concerned about mixing depth, and it's something we'll study, and in other lectures we'll actually go over ways to calculate the mixing depth in the photic zone. So light is very important, obviously, to these photosynthesizing organisms, and it's attenuated with depth in the sea. So on the left there you can see depth on the left hand side with the radiance, how much light, on the right, and you can see that it drops off very quickly with depth. It drops off exponentially. Now this varies depending where we are. Coastal waters around SAMs, temperate waters like this, sometimes places like Lough Creran you're only going to get the water penetrating 4 or 5 meters into the water. The Sargasso Sea, which is out in the big oceanic gyres, a lot of seaweed gathers there, water can get down to about 90 meters, and if you go into the big oceanic gyres where you get clear tropical water, you'll actually be able to detect light down to about 300 meters. So light is made up of different wavelengths, and it's absorbed differently in the water. So light is absorbed by the water molecules themselves. It's also absorbed by the biology in the water, the phytoplankton, and it's reflected and scattered by little bits of marine snow, particles, detritus, that kind of thing. So what we find is that infrared, red and ultraviolet, they get absorbed very quickly. They don't really penetrate into the sea at all. Yellow gets a little bit further, green and violet get a little bit deeper than that, but the one that makes it all the way down is blue. And that's why the sea tends to look blue if we ever go diving or snorkeling or whatever. When you look around, it all looks beautiful and blue. And that's because that's the only wavelength that can really make it down there. So light that we're going to use for photosynthesis we call PAR. So that's photosynthetically available radiation. And it lies between wavelengths between 400 and 700 nanometers. And the chloroplasts, the chlorophyll in the chloroplast is often different. There's different types, A, B, and C, and it's more reactive for different wavelengths. And there's also accessory pigments like carotenoids, which also absorb light at different wavelengths. And it just gives organisms a better chance of gathering any available light that's down there. So we tend to classify water into different types. So 1 through 3 are clear oceanic waters, 1 being the most clear, so that would be these big tropical or big gyres out in the Central Ocean. 4 tends to be clear coastal water. So we've got to put all of that together, and we've come up with a sort of simplified pattern of phytoplankton growth in temperate coastal waters. It's slightly different in the tropicals, in the tropics. So in the winter we have big convection zones where the water is overturning. We get lots of mixing, there's lots of winter storms. This brings nutrients up from the sediments on the bottom and mixes them through the water column, but there's very little light, so the phytoplankton can't grow. As we get into spring, we reach a threshold where there's enough light for them to grow and there's plenty of nutrients. So the phytoplankton grow exponentially, and they just bloom, and you get this big peak in phytoplankton. This corresponds with this sort of stratification of the surface layers. So you get these shallow layers that the phytoplankton get trapped in, plenty of light, plenty of nutrients, boom, off they go. This only lasts for a week or two. They quickly use up all the nutrients, they get attacked by viruses, and the population crashes and drops back down to zero. As we progress through the summer, the dinoflagellates, they tend to be a bit slower in growing. Because they've got flagellum, they can move, they can dip in and out of the nutrients through the picnic line. So they grow slowly through the summer and reach a peak in the middle of summer. And they love very stable, stratified water conditions. As we get into autumn, we get more mixing. There's some autumn storms. There's still, so that brings up more nutrients. There's still enough light around, so we often get another little bloom of phytoplankton. And then we go back into the winter again, water cools down, overturning, and the whole thing carries on. Now this is incredibly simplified. So what in real terms happens is that any time we get a storm, it can be quite regular in Scotland, if we get a storm, it mixes up the surface waters, it brings up nutrients, and then a couple of days later it's nice and sunny, the water stratifies and you get a big bloom of phytoplankton again, just depending on whatever was there. So in real terms, this would be a spiky, the green graph would be a spiky one. So you'd have little peaks of phytoplankton. Nothing like the spring one, but you get little peaks throughout the year. So this means that a lot of the work you do, if you're studying phytoplankton or primary productivity, involves working with stratification. So that's what we're really interested in. So here's just some pictures I've taken on cruises, showing the kind of equipment we're using there. So on the top left, that's a CTD, so it's conductivity, temperature and depth, and it often does chlorophyll and light dropping down to the water, and it's sitting below a carousel of water bottles, and these are 20-litre water bottles. So what happens is we drop that down through the water column, it gives us a trace on the computers on board the ship. People decide what depth they want to take a water sample from, and as the CTD is brought back up through the water column, the operator fires a button and it closes bottles at various depths. Once it's back on the ship, you find your bottle with the number that you wanted, at the depth you wanted, and you suck off the water and you take it away for analysis. In the middle here, this is Dave McKee from Strathclyde University. He works with light, he's a physicist, and the rig he's got is a self-built rig that monitors the light field, in this case under the ice. And in the bottom left, this is down in the South Celtic Sea. This is a Slocum glider they're just deploying there, and this effectively is like a CTD, but it's piloted from people back in the lab or on the ship, and it works with density, and it can glide down through the water columns, it dips and rises, and it can be deployed for several months at a time. Okay, so that's all for this introductory lecture. We'll have a Q&A session in a few days, so watch Brightspace, and I'll tell you when that's going to be. So go over this again, and I encourage you to look at the PowerPoint slides that I'll put up on Brightspace. Sometimes I have a few more slides than I've got here, and sometimes there's more extensive notes attached with them. There's also a series of videos to accompany this, which give you a bit more background information, and there'll be some papers you can look at as well. So the next time I meet you in the class, we'll have a Q&A session, so you can ask questions about this, and we'll see how things go. Okay, so thanks for listening, and bye for now.